Okay, so yes. So today class, I'll be talking about neurogenesis, how the neurons are formed, and I'll be covering three model system, specifically the lower one, the round one, C. elegans, and also I'll be talking about the favorite model system of mine, the Drosophila and Vertebrate, and also we'll back to the mammals, uh, the mouse model, and also the monkeys. Some studies also in the monkeys, the ferret. So we'll go one by one. So first we'll talk about how neurogenesis happens in round one, the C. elegans. So the C. elegans work was extensively studied. Mostly the lineage analysis were done by three famous scientists, Sidney Brenner, John, and Bob. So they got for their work on C. elegans how neurons are formed. Because C. elegans is a very simple organism, there is very few neurons. You can easily manipulate each neuron, and you can assess the function. So it is a great model organism to study developmental biology. So they did the work for the lineage analysis, and they got Nobel Prize in 2002. So according to Sidney Werner, he states that according to him, the science is the progress in science depends on the new techniques and new discoveries. You have new techniques, then you can discover something novel, and you can generate new ideas, and that happens probably in this order, like new technique, new discoveries, new idea. So according to him, so this is a famous quote. I just kind of impressed with this kind of quote, that's I put it here. So we'll go to the next slide, talking about how the neurogenesis happens during different developmental stages of C. elegans. So this is C. elegans starts with a P0. It is the fusion of sperm and ovum, egg to form the zygote. The P0 is the zygote stage. Now the zygote divide asymmetrically. So it forms a bigger cell, which is AB blastomere, forms by asymmetric division, and a smaller cell, which is which form the germline cells. Okay, this is the cell stage. Now it divides into four cell stage. So AB divides into ABA and ABP. Then P1, the germline cell, divides into EMS cell and P2 cells. You have 4 cell stage. Then you have 8 cell stage, 12 cell stage, something like that. Then AB blastomere again divides into the other AB blastomeres, and the EMS divides into MS and E. P2 divides into P3 and C. Again, in the next stage of the development, P3 divides into P4 and D, and P4 is the germline cells. It forms the germline. So during this process of gastrulation in C. elegans, the AB blastomeres, they come outside and form this epidermis and the epidermal layer, the ectoderm. They form the ectoderm, which consists of hypodermis and nervous system. And the EMS, the P1 cell, the progeny from the P1 cell, the EMS, MS cell, and E cell or C cell, they form the body muscles, somatic gonad, and more exclusively the P4 cell, it forms the germline. Okay, so here this is the next stage of the development which I talked about in the previous slide. You have the AB blastomere. It comes outside from the ectoderm, which consists of the hypodermis and nervous system, and other cells like E cell or other cells that come inside of the embryo and form the other internal organs. So this is another in the matured gastrula stage. So you have generation of endoderm, mesoderm, and also initiation of the other parts of the nervous system, like you have the generation of hypodermis and ventral central nervous system or the peripheral nervous systems. So that's how the gastrulation happens from single P0 cell to different types of cell. Then you have outward movement of cell, The epiblastome from the hypodermis or ectoderm, then other cell form, other organs. And here, this is the developed roundworm, which has different structure. Like you have ovary, you have this is a mature worm, which have intestine, and you have eggs. So this is the cross section of showing these animals, like the developed C. elegans, showing different organs like gut. You have endoderm. You have nerve cord in the either side, the dorsal nerve cord, the ventral nerve cord. You have the body wall muscles. So a typical lineage of in C. elegans undergoes 5 rounds of division to generate 9 neurons and 10 hypodermal cells. So it undergoes 5 rounds of division. So you can calculate here: first round, second round, third round, fourth round, and 5th round. So it undergoes 5 rounds of division to generate 9 neurons, which are shown in the red, and 10 hypodermic cells. So this is any question on C. elegans development? Okay. We'll go to the next section. So about Drosophila, and we already talked about Drosophila gastrulation. And here in this section, we'll be talking about how neurogenesis happens during the development of Drosophila, during the developmental stages. So this is, after formation of egg and sperm, they fuse and form the zygote, and zygote develops into blastula. Blastula develops into gastrula. Okay, so this is a matured embryo. I'll go mostly. So I'll come to this slide, then I'll go to the previous slides. This is already I have talked about. So here you have the dorsal side during gastrulation, and this is the dorsal side. So accumulation of DPP. DPP, a dorsal protein. It specifies the dorsal side of this during the development, and you have accumulation of other signal in the ventral side. Like you have a SUG, accumulation of SUG. So I will be talking more about the SUG, and twist. It specifies the ventral differentiation. So there is a gradient that express so you have DPP gradient more in the dorsal side. It goes and the gradient become very weak to the ventral side. Similarly, the SOG gradient is higher in the ventral side. It become very weak in the dorsal side. So this is just to show that different protein accumulate during gastrulation, and specifies dorsal and ventral side during the development of Drosophila. During the development of Drosophila. Okay, so yeah, so the neurogenesis happens because of the expression of SOG. I talked about SOG before. Expression of SOG RNA in the next slide. I talked about you look at this one. So here I talk about SOG, accumulation of SOG in the ventral side. So neurogenesis happens. The neurogenic region is defined by the expression of short gastrulation or SOG. Okay, so what happens is during gastrulation phase, the specific type of cells delaminates from the ventral side of this in the gastrula stage. This is the delaminating cell. It delaminates. Okay, and these cells are called as neuroblast cells. Okay, so here the neuroblast cells, and the neuroblast cells, basically the SOG defines the expression of the SOG actually defines the neurogenic region of this embryo. Okay, and it delaminates from this ventral side, and this called as neuroblast cells, and this neuroblast cell divides into divides asymmetrically, forms 2 different cells. It forms one bigger cell called as neuroblast cell, another cell called as ganglion mother cells. Okay, so again this is one ganglion mother cells, then again the neuroblast divides from second ganglion mother cell, then neuroblast divides to forms third ganglion mother cell. So each time the neuroblast divides to form one asymmetrically divides to form one ganglion mother cells and a neuroblast cells, and the neuroblast cell actually has is a stem cells. So it can actually has a capacity to generate another ganglionic mother cells. So that's how you have expression of SOG, then the generation of a neurogenic region in the ventral side, and formation of a neuroblast. Neuroblast divides to form a ganglion mother cell and again neuroblast cells. So this already we talked about. So the neuroblast cells encodes. So the next question is, what is there in the neuroblast cell which is specifying or which is actually generating this neuron? So the neuroblast cells express pro-neural genes. Okay, and this is a complex of there are a lot of genes expressed in the neuroblast. So you have 4 group of gene that is expressed in the neuroblast, which is one group contains 4 different genes that is called as Akethy-Skute complex, ASC complex, which contains these 4 genes, Akethy, Skute, and essence and lethal of skute. So these are the pro-neural genes which expressed in the neuroblast, which defines the neuron. Okay, and those genes basically forms a specific type of transcription factor called as basic helix loop helix, BHLH family of transcription factor. Okay, so if I kind of summarize this, so you have delamination of the cell because of expression of SOG, and once the cell delaminate, it forms 2 asymmetrical divides from 2 cells, one bigger cell called the neuroblast cell, another ganglion mother cells. Now those cells contain those pro-neural genes, 4 group of pro-neural genes, which is called as ASC complex, which contains Akethy, Skute, essence, and lethal of skute. And that actually codes for a specific type of transcription factor, which defines the neuronal cell fate. That transcription factor is basic helix loop helix transcription factor. Okay, so here this is an immunostaining has done in the Drosophila embryo, showing the expression of pro-neural clusters. So each cluster or each hemisegment contain 2 clusters. So you have 1 cluster here, 2 cluster in one hemisegment, another hemisegment, 2 cluster. So you have expression of, so it express throughout the body. This is called as pro-neural clusters, which is shown here. And initially the clusters will express ASC genes, and which further develop into, which in further development, one cell actually continues to do so. Okay, so this is how you have expression of the pro-neural clusters in Drosophila, which specifies or which generates the neurons because of the expression of the BHLH transcription factor, basic helix loop helix transcription factor, which actually transcribes the genes required for the neuronal development. Okay, so any questions? No? Okay, so now the next question is, which cell become neuroblast? How there are a lot of cells. There are many neuroblasts. Then among that, one cell should actually one cell should differentiate into neurons. So right, because you have many neuroblasts during the development, asymmetric division, you have many neuroblasts. Then among all the neuroblasts, which cell actually take the center stage for the development for the neurons? So, and from the past, a lot of studies have been done to identify actually among bunch of neuroblasts, which neuroblast taking the function of becoming a neuron. So one experiment people have done, that is the laser ablation experiment. When the cell is delaminating, okay, so they treated this, and this is the gastrula stage of the embryo. So that what they have done is they have treated the cell or irradiated the cell with laser, and they have cleaned this, so killed these cells by the treatment of laser. You can see that you do not have formation of neuron. So, and each neuroblast which is delaminating from this ectoderm actually again gives signal to the neighboring cell, which can also become another neuroblast. So when the laser ablate these cells, you do not have formation of neuron. Also, the neighboring cells also are not being, they are also not developed at neuroblast. So this is one experiment to suggest that, okay, by, if you treated this, you irradiated laser with a delaminating neuroblast, you can kill the neuron actually. You can inhibit the expression of neurons. Okay. So the next experiment that people have done, they have identified a set of genes which is required for the development of a neuroblast. Okay. So the two sets of gene studies have been done from the past. One is this gene called as Akite loss of function mutant, and also the Notch delta loss of function mutant. When they mutate this complex, Akite complex or Notch delta pathway, they found that the neuroblasts are not formed, and they say that these are the factors required for the neuroblast development in Drosophila. So quickly, I'll go through the delta Notch signaling pathway and how it specifies or defines the neuroblast in Drosophila. So this is a normal scenario, and this is another scenario where you have reduced expression of delta and in the suppressed cells. So here you have there are two cells, one cell expressing delta. Delta is the ligand, and this is an intracellular kind of transmembrane ligand, and you have a notch in another cell. So this is the outside of the cells. So delta ligand bind to the notch receptor, and after binding to the notch receptor, it gets activated. Okay. So then what happens is you have expression of gamma secretase enzyme because of the activation of the pathway, and the gamma secretase has a proteolytic activity. It cleaves this intracellular notch domain, and those intracellular domain release after cleavage, and it translocates in the nucleus. It binds to this enhancer of split, and that in turn binds to the promoter of this AAC complex, and which transcribe the gene, which transcribe the gene, and also it forms a neuron. So as I mentioned before, there are 4 sets of gene actually required for the development of neurons or proneural genes. One is the AAC complex or Akite complex from the previous slide. So one of these is Akite complex, which is activated by delta Notch signaling pathway. So in another scenario, what happens is when you have a reduced expression of delta, so delta is not sufficient, and that's why the notch is not get activated. So you do not have the intracellular domain cleaved. You do not have the intracellular domain which need to be cleaved, and also the enhancer of a split is bounded to the intracellular domain of this one, the notch receptor. So this is not free also. So this is not free. You have reduced expression of delta, so that the other other round of binding will not happen, because this complex is not free. So it remain bound to this promoter. It is not free. So that's why you have suppression of transcription. So because of reduced expression of delta, you have suppression of transcription. You have suppression of transcription of this complex. So you do not have expression of the proneural genes. That's why you do not have a specification of the genes required for the neuron development in Drosophila. Any question? Okay, so let us talk about what is the model for neuroblast development in Drosophila. We'll be talking about the delta Notch signaling pathway more. So as I mentioned earlier, you have expression of proneural clusters. You have expression of genes in the proneural clusters, which defines the neurons. Okay, then there are many cells in the proneural cluster. Then which cell become neuroblast? That is also important, right? So among all these cells, among all the cells, only one cell express larger amount of AAC complex and this transcription factor, the basic loop helix transcription factor, which again turns on delta expression. So among all these proneural clusters, one cell dominated and expressed more of these. Okay. So here you can see that it dominated and expressed more of this AAC complex, expressed more of this transcription factor, BH-LH transcription factor, and activates delta. Okay. Now, what happens is, after activation of delta, it activates in the surrounding cell, which expresses the notch. Okay. So after activation of notch, you have expression of you have the signaling from the delta notch, then you have expression of these genes required for the transcription. So this type of activation is called as, lateral inhibition. This type of thing is called as lateral inhibition, which determines which cell become neuroblast. So among all these cells, one cell dominated and express lot of transcription factor, ASE complex, also which turns on delta. Okay, now you have expression of notch. Then, so this cell become dominated and other cell remain suppressed. It suppresses the other cells, surrounding cells. Then you have expression of the genes required for the neuronal development. So that's how you have because of the lateral inhibition, one cell becomes dominated and defines, it forms a neuroblast, and it forms the net set of neurons required for the development. Any questions? Okay. So this is how it happens. So you have proneural clusters. So I'm showing the two cells here, we zoomed here. So you have this complex called as ASE complex, which expresses these molecules, that is mass and neurogenin molecules, and these molecules further activate the delta, and delta binds to the notch of another cells. So one cell communicate to other cells with notch. Now these cells also express delta. It can bind to the notch. So the two cells communicate each other through delta notch signaling pathway. Now among these, one of the cells become, one of the cell in the proneural cluster become neuroblast with this mechanism. What happens is these cells become neuroblast, and these cells remains as non-neuronal cells. So how it happens? So you have this ASE complex which expresses these genes like mass and neurogenin gene, which activate delta. Delta binds to notch, and after binding to the notch, the other cell suppress of hairless, and these activate enhancer of split protein, which inhibit the ASE complex of other cells. Now you have a reduced expression of delta. So because of reduced expression of delta, this cell become non-neuronal and this cell become neuroblast. Okay. So with this pathway, this cell express more delta, it becomes neuroblast, and it inhibits the other cell by this mechanism to become non-neuronal cell. So this cell become non-neuronal and this cell become neuronal, the neuroblast. So because of expression of delta, you have specification of neuroblast. Okay. So it's kind of this will become non-neuronal, this will become neuronal precursor, the neuroblast. So this is here shown here, the same thing. So you have reduced expression of delta in other cell because of this pathway, and acutase QT protein actually express BH-LH transcription protein factor, which actually becomes neuronal precursor, the neuroblast. So that's why, because of expression of a transcription factor, one cell become neuroblast, and it expresses more delta, and the other cell become non-neuronal cells, and because of expression of less delta. So this is how the neuroblast form in Drosophila, and you have generation of neuron in Drosophila. Okay. Any questions in the Drosophila section or C. elegans section? So the take-home message is the by combinatorial action of ASE complex delta notch pathway, you have generation of neuroblast in Drosophila and generation of neurons in Drosophila. Now we'll go to the next section, that is the mammals. So it is more interesting to know. So in the mammals, the neural tube during the development, the neural tube contains progenitor for the neurons and glia. So this is a cross section of a developing chick embryo, spinal cord, derived from the neural tube, and this takes around 6 hours. So this is the lower panel is the time lapse image showing the development of the spinal cord neural from the neural tube in the chick embryo. So what happens during development of what happens during development of neurons in chick embryo spinal cord? What happens is it is so first some special type of cell present in the ventricular surface. So you have in the neural tube, you have the ventricular surface, and the progenitor cells for the neuron and glia are present in the ventricular surface of developing neural tube in the chick embryo. Okay, so those cells, these are the bipolar cells. So those cells show in and outward movement. So and you can see that here the cells present in the ventricular surface are at different stage of the cell cycle. So you have the S phase, the G2 phase, and the M phase, and G1 phase, and those nucleus in the cells show inward movement from S phase to G2 phase. Okay, so you can see that in the nucleus which are now in the outer side of this ventricular surface, you can see that in the G2 phase they move inward, and they remain in the G2 phase. They forms the mitotic phase, M phase, and they again move outward. Okay, so because of the inward and outward movement, they actually divides to form the neurons. So you have progenitor, the neuronal progenitor present in the ventricular surface, which contains the progenitor for neuron and glia, and these cells actually show inward and outward movement to generate the neurons. Okay, so this is a time-lapse image showing that the inward movement and outward movement of a neuronal progenitor in the cheek spinal cord the cheek spinal cord. Okay, so now how to study the which cell becoming neuron, which cell becoming which type of neuron. So you can do that by fat mapping using retrovirus. So what you can do is you can clone specific type of gene, and you can track actually throughout the development. You can use the beta galactosidase genes, and this actually cleaves a substrate to form the blue product. Now what you do is you took those progenitor cells, which is in the ventral surface of the spinal cord, and inject into one of the progenitor cells. So the virus have the beta galactosidase gene, so it injected. Now those viral genome again integrate to the progenitor genes or progenitor genome. So during the division, the virus also those genome also get integrated and divides to form other cells. So other cell contains the virus genome. Now if you treat the substrate, because some of the cells will contain the beta galactosidase gene, right? So if you treat that cell with the substrate, it forms the blue product. Now you can label that, okay, this cell become blue, and you can track those progenitor cells becoming microglia or oligodendrocyte, something like that. So by this technique, you can actually heat map different type of neuron developed from the progenitor cells. So you can see that some of these progenitor cells become motor neurons, some of them from the oligodendrocyte, some of them from the astrocyte. So you can track the cells by using this technique, like which neuroblast forming motor neurons or forming astrocytes, something like that. Or another technique you can use to basically track the neurons. So one is called as radio-labeled thymidine, or you can use bromodeoxyuridine or BRDU or EDU. So basically those modified bases actually bind to these nucleotides, binds to these highly transcribing cells or highly dividing cells, so get incorporated during DNA synthesis. And so here, this is an experiment people have done at different stages of the mouse development. So you have E11 stage, you have E13 stage, you have U15 stage. You want to know at which stage of development, which type of neuron is formed. So if you inject at E11 stage with radio-labeled thymidine, you can basically track the tissue, the neurons which are found in the earliest stage of the development. Or you can inject that in E13 stage, you can see the neuron which are from the later stage of the development or very later stage of the development. So by this way, you can actually study what kind of neuron is being formed at what stage of development, which will be more clear here. So here, the pregnant female injected with radio-labeled thymidine, so injected at E11 stage or E13 stage or E15 stage. Then you take the take the tissue and do the autoradiography. You can actually visualize different type of neurons which are generated different stages of the development. So E11 stage, you can label the subcortical white matter, or E13 stage, you can label layer 5 and layer 6, the deep cortical layer you can actually label, or at E15 stage, you can also label layer 4, 3, and 2. Okay, though those are formed at the latter stage of the development, the E15 stage of the development. Any questions on fat mapping, neuronal labeling? This is just to show that you can actually track the progenitor. It is actually forming which type of neuron at a different stage of the development, either by retrovirus or by using the radio-labeled like thymidine or bromouridine or uridine. Okay, so we'll move to the next one. How neurogenesis happens at the ventricular zone of the neural tube. As I mentioned earlier in the earlier slide, that the neuronal progenitor present in the neural tube, in the ventricular surface of the neural tube, okay, which show in and outward movement, to generate different type of cells. So first it show the inward movement, and it go to the G2 phase, then M phase, then it come to the S phase. So that's why the you have generation of progenitor and generation of different kinds of neuron. So we'll go that with detailed mechanism, how the neurons are formed in the ventricular zone. So this is a typical, this is again taken from the part of from the telencephalon. So this is showing the part of the telencephalon have different layer. So you have ventricular zone, which I talked about before. You have subventricular zone, you have intermediate zone, you have cortical zone and marginal zone. Okay. So the progenitor are actually derived or in the ventricular zone. So in the coming slide, I'll be talking about mostly the progenitor for the neurons are they are generated from the radioglial cells, glial cells. So we'll be talking about more about this in the coming slide. So here, this is a typical structure of radioglial cell and neuronal cells showing in the intermediate zone. So you can see that this white thing that you are seeing is the process for the radioglial cells, and the blue one is the migrating neuron. Okay, so it is generated in the ventricular zone. It migrates to the intermediate zone, then it enters and it goes into the cortical and marginal zone. Okay, so so there are distinct process. So you have the process of radioglial cells. This is the process for the neuron. This is called trailing process, and this is the migrating neuron, and this is the leading process of the neuron. Okay, so basically the radioglial cells act as a support for the migrating neuron. Now, so here, this is a typical structure of radioglial cells and migrating neuron, which is generated from the radioglial cell. We'll be talking about this. So here at the end of the radioglial cells, you have you have end foot, and also it releases a specific glycoprotein which helps in the migration of the neuron. Okay, so so Nocta showed that the radioglial cells are the neuronal progenitor cells. As I mentioned before, the neuronal progenitor cells are developed from the radioglial cells. The radioglial cells are mother for the neuronal progenitor cells, and also it helps in the migration of cells, neuronal cells. Okay, so here, this is the experiment that has been done to show that radioglial cell actually are the progenitor for the neuronal cells. So this is the radioglial cells. Okay, the radioglial cells asymmetrically divide two cells. One is the larger cells, is the daughter cell for radioglial, and this is the daughter cell for the neurons. So these cells, the daughter cell of the radioglial cells again divides to form two cells and it forms the radioglial cells, so that it can again form next set of the neurons. So the neurons, so the other cells, the smaller cells, becomes neuron and it migrates to the cortex, which is shown here. Okay, so this is one mechanism. Like you have formation of radioglial cells which act as a progenitor for neural cells. It asymmetrically divide from two cells, one bigger cells which remain as radioglial cells for the daughter radioglial cells. The other smaller cell become neurons and migrate to the cortex. Okay, so another mechanism is that so you have two kinds of division happens in the radioglial cells. One is the asymmetric division that happens in the ventricular zone, and you have symmetric division that happens in the subventricular zone. Okay, so here what happens is the radioglial cells divides to form asymmetrically divides to form two cells. One is the daughter cell for radioglial, another is the neuronal progenitor cells. Now the neuronal progenitor cells in the subventricular zone divide symmetrically from two neurons, and the two neurons migrate to the cortex. Okay, so you have two kinds of division happens. One is asymmetric division and the symmetric division. Asymmetric division happens in the ventricular zone or ventricular surface, and symmetric division happens in the subventricular zone. Okay, any questions? So here is the model and also showing symmetric and asymmetric division. So here, this is the radioglial cells. It divides asymmetrically, kind of summarizing, forming radioglial cells, daughter radioglial cells, and progenitor for the neuron. Another neuronal cells. The neuronal cells migrate. In one case, it migrate to form to the cortex, and another scenario it forms divides to form two cells. And the daughter cells in the subventricular zone symmetrically divides to form two neurons, and they migrate to the cortex. So the neurons are formed by the asymmetric and symmetric division. Okay, asymmetric division happens at the ventricular zone, and symmetric division happens in the subventricular zone. So here, this is the chart showing percentage of asymmetric and symmetric division in different surface. Like ventricular surface, you have mostly the asymmetric division which is dominated, and the subventricular zone you have symmetrical division more. Okay, this is just to summarize. So the take-home message is you have to remember that the subventricular zone it shows the symmetric division happens in the subventricular zone, and asymmetric division happens more in the ventricular zone. Okay. This is again the summary, and finally after the division, the radioglial cell transform into astrocytes. The final final division of a radioglial cell is the transformation of astrocytes. Okay, so you have formation of neurons from the radioglial cells, and after that, finally the radioglial cell transform into astrocytes. So this is how the neurogenesis happens in the ventricular zone and subventricular zone of developing mouse embryo. And this is the then we will be talking about how the things are organized in the cortex. And the cortex, the cells forms in the ventricular zone and migrate to the cortex. And the cortex is a multi-layer structure. Lot of cells, it receives different processes from different parts of the brain. So you have like layer 1, 2, 3, 4, 5, 6. Okay, and each layer receive different projection from different parts of the brain, which is kind of I have written here like layer 1, which type of processes receive layer 1, layer 2, or layer 3. All these things are mentioned here. Okay, so this is just you don't have to remember this slide. I just want to show that how complex is the cortex. So cortex is a complex layer. It receives inputs from different parts of the brain. Okay. Now, again, you do not have to remember the slide. So here I am showing the complexity of the cortex in different organisms. The human, how the cortex is complex. It has 6 different layers, and the same thing is there in the rat and the mouse. So this is showing that the cortical thickness of human is around 2,622 micrometer. It is again in the rat, it is 1,827 micrometer. You don't have to remember, and you just want to show the complexity of cortex. It's much more in the human than the other model system like mouse and rats. Okay, so here the human content, the human brain has the symmetric synapse and asymmetric synapse. So the cortex has 89% asymmetric synapse, and the asymmetric synapse are having narrow synaptic cleft. Okay, and the asymmetric synapse having the synaptic vesicle are spherical in asymmetric synapse, and the symmetric synapse, they have narrow synaptic cleft. Okay, so there is more asymmetric synapse in the human as compared to synaptic synapse. So again, in the mouse or in the rat, you have more asymmetric synapse. Okay, and you have more number of synapse in human. So everything is, you don't have to remember the slide, just to show that the complexity of a cortex. Okay, so coming back to this slide, so development of cerebral cortex, in the cortex, you have the cortex is receiving input from different parts of the brain, and also the cells also migrate to the cortex. It shows the migration, differentiation. So the program that described in the previous slide generally applied throughout the developing CNS. Okay, the previous slide I was talking about, the program required for the developing CNS, both cortex, different layer of the cortex. But here I want to highlight that the cortex that I talked about before, that contain the visual cortex and also different region of the cortex. But here I want to, you want to study, the neuronal fat in different layer of cortex. Okay, so you can do that by, again, the same thing, by using the fat mapping, or the same thing by thymidine injection, radio level thymidine injection. You can do the analysis in different part of the cortex. So if you inject at E45 of the monkey visual cortex, you can actually label the cells which are generates, you can label the neurons which are forms in the earlier stage of the development, like the deeper layer, the layer 6. Okay, if you inject radio level thymidine E62 days, you can actually label layer 5. So this kind of experiment people have done to study different layer of the, because cortex is a multi-layered tissue. It is a multi-layered structure. So if you want to study the different neurons or different fat of the neurons in the cortex layer, you can do that by injecting the radio level thymidine at a different stage of the development. So if you inject in the earlier stage of development, you can study the deeper cortex layer. If you inject in the latter stage of the development, you can study the surface cortex layer. Okay, so this is just to show that you can study those brain section in the deeper layer by doing this experiment, by injecting radio level thymidine. So here, just want to show how the cortex layer is formed during different stages of the development in a monkey, during the development of the brain in the monkey. So here, this is the earliest stage of the development, this is the mid stage of the development, and this is the latter stage of the development. So here, this is defined in different regions, the cortex layer. You have ventricular zone, you have intermediate zone. The ventricular zone have the new progenitor, which forms the neuron and also the glia. And then after formation of a neuron, they migrate to the different layer. So at the earliest stage, you have formation of intermediate layer, the subplate layer. So there is a temporal specification of cell fate. So which cell will form at what time? So that depends on the developmental time. So in the earliest stage, you have formation of cortical plate, subplate, intermediate plate, and ventricular plate. Okay, then because of the in-out generation of cortex, in-out means you have, the cells which are formed in the ventricular surface, which will come out, as I shown in the very first slide. Like the cell go into S phase, the M phase, then the mitotic phase, the G phase, M phase, then you come to the S phase. It shows like inward and outward movement. Because of those, because of this phenomenon, you have generation of neuron, and the neurons are actually moving outwardly, migrating to the cortex. So after some time, you have development of subplate, layer 5, so layer 6, cortical plate, marginal zone, and finally you have generation of all these layers. So you have formation, you have generation of different layer with time. You have out movement of the cells, and you have migration of cell to the final destination, final location, to the cortex, different part of the cortex. Okay, by this mechanism, you have generation of cortex or formation of different cells in different part of the cortex, and those things are actually guided by radioglial cells, which are actually formed in the ventricular zone. Okay, those are the progenitor for the neuron. So first, the radioglial cells are formed in the ventricular zone, and they divide to form progenitor for the neuron. Now at a different, now you have generation of a different layer. Okay, you have in and outward movement. Outward movement of the cells. Then you have migration of cells. Okay, now you have generation of cortex. Got my points? And finally, the radioglial cell differentiated into astrocytes. So that's how you have generation of cortical layer, different layer of cortex, and that depends on the time. Okay, which stage of the development, you have which type of layer will develop. So those things are studied by using, radio level thymidine experiment. So E15 stage, you have generation of one kind of layer. E25 stage or E30 stage. So you have different time point where different layer are formed. So that is called as temporal development. Okay, it depends on the time. Then you have outward movement of the cells. You have migration of cells, and those things are guided by the radioglial cells. And finally, the radioglial cell transform into astrocytes. Okay, so that's how you have generation of cortex. And also another mechanism is that it depends on temporal gradient of. It also depends on different temporal gradients. Okay, so neurons are formed different. It depends on the what time neurons are forming. It also depends on different locations. So you have different locations where the neurons are formed. So you have here there are three mechanisms which are discussed. One is the reverse rostrocaudal gradient. One is the lateromedial gradient. You have ventrodorsal gradient. So in the brain, you have different surface. You have ventral surface, you have dorsal surface, you have lateral surface, you have medial surface, you have rostral surface, rostral side, you have caudal side. So what happens? First, the spinal cord is generated, then the rostral side is generated. Rostral side is the brain, and the caudal side is the spinal cord. So this is called a reverse caudal gradient. It depends on the parts. It depends on which part of the brain. So there are a lot of things happens here in the brain development. It depends on the time. Also, it depends on the location. Okay. First, the caudal part will be generated, the spinal cord. Then the rostral part will be generated, the brain. In the lateromedial gradient, first the lateral part of the brain will be developed, and then the medial part will be developed later. Then the ventral first, in the ventrodorsal gradient, you have development of the ventral side first, then you have development of dorsal side later. So it depends on the time, it depends on the site, like the location of the brain. So that's how you have neurogenesis happens in the brain, and you have development of the brain. And the last mechanism that I want to discuss is it depends on the local environment. Okay. So this is again done in the ferret model system. So this is E29 stage of the development of the brain, embryonic stages. And this is the postnatal, the day one after birth. And this is the adult stage. So what people have done is they want to know how the local environment affect neurogenesis. Okay. So here what they have done, they have taken cells from the ventricular zone at the E29 ferret embryo, and they have transplanted that to the P1 stage of ventricular zone. Okay, so when they transplant these cells to the ventricular zone of P1 stage of the ferret, they found that these cells become these cells keep the older cell fat. Okay, the fat transformed by the newer you can see that the younger cells which are transformed from earlier stage of the embryo, they are becoming the older cells, because some kind of signal that is secreted from the older, like P1 stage, which transforming the newer cells, which is transplanted from E29 to P1, those cells actually becoming the older cells, because you have something getting secreted from the ventricular zone, which is actually inducing those younger cells to become older cell fat. That means it depends on the local environment. Okay, the neurogenesis also depends on the local environment. Now, another experiment people have done, where they have taken the cells, the ferret at the E29 stage from the ventricular zone, and they have co-cultured with other cells in this region. So they have taken, cells from the ventricular zone, okay, and they co-cultured with other cells from the E29 stage. That is called as pellet cells. So when they co-culture with the pellet cells from the E29 stage and transform that the transplant to the P1 stage of the development, the P1 stage, you can see that they becomes the younger cells, because those cells which are those removed from the E29 stage, receive signal from the younger cells here. So other cells from the E29, they receive signals from the younger cells. Now they become younger cells. So the pellet actually fixing the cell fat. So it depends on the local environment. That's why I want to highlight that it depends on the local environment. Here, the local environment is the older, the P1 stage, the postnatal 1 stage. So the local environment is older here. That's why even you are putting the younger cells, they are not behaving as a younger. They are behaving as a older. Now, here you are co-culturing with other cells, other younger cells. Now the cell become younger. So the fat become younger. Even if you transplant to the P1 stage or P1 stage, they remain as younger because the fat is already resided here from the younger cells. Now, the last experiment that what they have done, they have taken the cells from the P1 stage, the older stage, and transplanted to the younger E29 stage. Here, it did not fat. The fat is restricted because this is already older. So even if you put in the younger embryo, it did not transform into a it did not show any fat. So it remains the same. That means the three things happening during neurogenesis. It depends on the expression of it depends on the expression of factor from the radioglial cells. Those radioglial cells secrete a specific glycoprotein which help in the migration, and it depends on the time, at what time which cell will develop. It depends on the temporal it depends on the temporal manner, and also depends on the local environment and the surrounding cells. So that's how you have neurogenesis in the adults. Neurogenesis happens in the developing embryo. So next class we'll be talking about some adult neurogenesis. So any questions from these slides, from this vertebrate section? Yes. Sorry? Yeah. So you will have question coming up to this today lecture. You have all this. So next lecture will not contain the questions. So up to this. So I have 22 questions from my 5 lecture, and the rest of this Ryan will come from the Ryan, 18 questions from the Ryan. So you have at least 4 questions will be from the each lecture. You'll have 4 questions. So all these questions are from the slide. So yeah. I just give him the overview of neurogenesis. If you want more detail, I will recommend the book Sense, or if you have any doubt, we can discuss more. But I just followed, the last year how they have covered the neurogenesis. Similar way I have covered. Yeah.